Right, welcome everyone. Thank you for joining us for the July 2025. Oh my gosh, it's not 26, it's July 2025 edition of Lunch a la Carte, which is a monthly Zoom call sponsored by the CART Fund with support from our Rotary Club of Alzheimer's Research. Everyone. Thank you for joining us for the July 2025. Oh my gosh, it's not 26, it's July 2025 edition of Lunch a la Carte. I apologize for that. I'm not sure if you guys could hear that or not, but okay. Sorry, I apologize. I'll go in and edit that out afterwards. I don't know what's going on. New computer. So we are proud to sponsor this monthly event with from the CART Fund, also in cooperation with the Rotary Club of Alzheimer's Research. And we love this opportunity to hear from one of our previous CART researchers every month. Just a reminder, you can always catch the replays of these on our YouTube channel, but you can also find them, the schedule for them in the DAC-DB calendar and on our website. And we would love for you to register in advance. Then you get the Zoom link via email. A couple of other events that are coming up. This Thursday night is the first of our monthly Zoom calls in the evening for our CART Fund volunteers. We call that CART Conversations. We hope to have lots of folks on the call for that. And that is usually on the third Thursday of every month. There will be a few months that gets altered due to schedule conflicts. But we do have another lunch a la carte already scheduled for Wednesday, August the 13th, and another one on Wednesday, September the 17th. And then there will be some others coming after that. So we encourage you to get signed up for those. It is my pleasure to introduce to you our speaker today, Dr. Michael Karin. He is a distinguished pharmacology professor at UC San Diego and a leading researcher in inflammation, cancer, and metabolic diseases. He graduated magna cum laude in 1975 from Tel Aviv University and got his PhD in molecular biology from UCLA in 1979. His groundbreaking work uncovered how certain genes and proteins link inflammation to cancer, especially in the liver and digestive system. And his lab was the first to show how conditions like fatty liver disease can lead to liver cancer and how the immune system plays a role in that process. He received a grant of $150,000 from the CART Fund in 2017 to explore how blocking a specific protein complex might help prevent or treat Alzheimer's and other age-related brain diseases. Dr. Karin, thank you so much for spending time with us today.
Well, thank you for inviting me, and most important, thank you for the precious support that was provided in 2017, and I'm glad you're continuing with that because I think scientists need more support like that from government independent foundation. So today I'll talk about some of the work that was supported by the CART Fund. And I apologize that some of the slides may be a bit complicated, but I'll try to simplify them. So I'm not going to go through a lot of the details. And I'll start with what we were doing in 2017, supported by the grant, and how we progressed later on to get some better understanding of some of the inflammatory diseases, including Alzheimer's disease, that are related to the NLRP3 inflammasome. So I'm doing share screen. And okay, do you see my presentation? Yeah. Okay, is my presentation visible?
Yes, sir, we have it. Thank you.
Okay, great. Okay, so my title is The Road Less Traveled, from Mitochondrial Stress to Alzheimer's Disease. So those of you who have some basic knowledge in biology know that our cells have some little organelles called mitochondria, which serve as the power plants that fuel our cells and actually fuel our life. through the production of ATP and different other metabolites. So mitochondria are very important for our healthy existence, but sometimes mitochondria can undergo stress, and then these stressed and damaged mitochondria are actually pathogenic because they release a variety of substances that can provoke inflammation. And this is a process that is believed to take place during what is called now inflammaging. We all know that as we get older, we are more likely to suffer a variety of inflammatory diseases. And unfortunately, old age can also increase the risk of neurodegenerative disease, Alzheimer, Parkinson, and several other related diseases. So we are interested to know how damage and stress mitochondria evoke all these pathogenic effects. So we and others found that mitochondrial stress results in the leakage of mitochondrial DNA to the cytosol and the extracellular milieu. So mitochondria actually originate from symbiotic bacteria that populated the cells of the very first eukaryotic organisms. And as a result of their microbial origin, they contain their own DNA genome, which is independent or still controlled by our own genome, which is present in the nucleus. And as mitochondria get stressed, they can leak this DNA, and I'll talk about the process that involves the DNA leakage, and elevated amounts of cell-free, circulating cell-free mitochondrial DNA, or CCF mtDNA, were reported in the elderly and in patients suffering from a variety of metabolic, autoimmune, and neurodegenerative diseases, including Alzheimer's disease. Inflammaging has been associated with elevated amounts of mitochondrial DNA in the circulation, but the actual origin of that DNA, where it comes from, is still a mystery. In most reports in the literature, the oxidation state of the circulating mitochondrial DNA has not been examined, but we found using macrophages that mitochondrial DNA release required its oxidation on deoxyguanine residues, and then it's mediated by a repair nuclease called FEN1, whose inhibition actually inhibits inflammasome activation. We therefore assume that most protein-free mitochondrial DNA found in the extracellular milieu in the circulation is oxidized to begin with. And importantly, as far as Alzheimer's disease is concerned, insufficient expression of a repair enzyme called OGG1 that specializes in the removal of oxidized residues from mitochondrial DNA is an Alzheimer's disease risk factor. And actually, there's almost a 40-fold increase in Alzheimer's disease risk in patients that have loss of function mutations in OGG1. However, none of these studies tells us whether oxidized mitochondrial DNA is a marker of inflammaging, neurodegenerative diseases, and so on, or is actually a trigger of the disease. And that's what we set up to do. And our own work is focused on the relationship between oxidized mitochondrial DNA and a protein machine called NLRP3 inflammasome. So the structure of the NLRP3 inflammasome has been solved, and we know that the NLRP3 protein, which is actually the sensor of cellular stress, somehow undergoes a conformational change. Then it can bind to another protein called ASC, which serves as a scaffold for recruitment of a very important enzyme called caspase-1. And caspase-1 is important because once it's activated in response to assembly of the NLRP3 inflammasome or other inflammasomes, it leads to the processing of pro-IL-1 beta to mature IL-1 beta and pro-IL-18 to mature IL-18. So what are IL-1 beta and IL-18? These are cytokines, which are molecules that are secreted by inflamed cells in inflamed tissue, and they lead to further activation of the immune system. So they can initiate and propagate inflammation. There are a lot of studies about different activators of the NLRP3 inflammasome, which include ATP, uric acid, which is involved in gout, cholesterol crystals, fatty acids, and amyloid beta fibrils, which again connected to Alzheimer's disease. And several other substances, including silica particles, asbestos, alum crystals, and so on. And it's been known for a long time that IL-1 beta is one of the key pro-inflammatory cytokines. It can affect many different cell types in the body, and it's been involved in cardiovascular diseases, liver diseases, metabolic diseases, and so on. But activation of the NLRP3 inflammasome can do a lot more than just produce IL-1 beta. And that's what I will describe to you next. So before I get into our own work, people have linked the NLRP3 inflammasome to a variety of diseases, including cancer. There are some specific inflammatory diseases called CAPS, cryopyrin-associated periodic syndrome, which are actually caused by mutation in the NLRP3 molecule. It's been linked to autoimmune diseases, which I'll spend quite some time talking about, such as lupus, different eye diseases, and most importantly for today's discussion, it's been linked to neurodegenerative diseases, both Alzheimer and Parkinson's disease. In about 10 or 15 years, actually almost 15 years ago, the laboratory of Jörg Chop, a good friend of mine who actually passed away about 10 years ago, who actually discovered the NLRP3 inflammasome, has shown that mitochondria are likely to be involved in activation of the NLRP3 inflammasome. And the substances that were shown to activate the NLRP3 inflammasome have very little in common. They are not related chemically or physically. And the question is, how can such a diverse array of signals and molecules, molecules inherent to our body, foreign molecules such as asbestos, can all lead to activation of the NLRP3 inflammasome? And what Jörg suggested is that they all impinge on mitochondria and lead to the release of mitochondrial DNA, which may activate the NLRP3 inflammasome. but they actually did not provide very clear evidence for this hypothesis. So with the funds from the CART Foundation, we started investigating the link between mitochondria and oxidized mitochondrial DNA with the NLRP3 inflammasome. And in 2018, we published a paper in Nature showing that newly synthesized mitochondrial DNA, which we label by incorporation of a base called bromodeoxyuridine, for which we have antibodies. So if the DNA reacts with these antibodies, we know that it's newly synthesized. And we also have antibodies that can detect the oxidation state of the DNA. So if you see a reaction with these antibodies, you know that the DNA is oxidized. And we have shown that if we pull out the ASC subunit of the NLRP3 inflammasome, we also pull out newly synthesized and oxidized DNA. We have used several methods to show that this DNA actually comes from mitochondria, including staining of cells with antibodies that show newly replicated DNA and show that its co-localization with ASC, which is this spot here. And what we have concluded in this study is that new mitochondrial DNA synthesis catalyzed by an enzyme called CMPK2, cytosine monophosphate kinase 2, which is specific to mitochondria, is required for the production of oxidized mitochondrial DNA and activation of the NLRP3 inflammasome. And the reason that new mitochondrial DNA synthesis is so important is because in resting cells, the DNA within mitochondria is tightly packed with proteins, and it pretty much looks like that. It's not exposed to reactive oxygen species, which there are plenty of them within mitochondria. But once you start replicating the DNA, you loosen up this structure called the nucleoid, and you start getting relaxation of the DNA. You strip the proteins from the DNA, and then you end up with totally naked DNA that can be attacked by reactive oxygen species that are produced during mitochondrial respiration. And older and stressed mitochondria produce larger amounts of reactive oxygen species, and that's why you see more mitochondrial DNA oxidation with older age and in different disease situation. We have shown that the oxidized mitochondrial DNA is fragmented. This is a method to look at the size of the DNA. And we found that the oxidized mitochondrial DNA in the cytosol is much smaller than the DNA that remains in the mitochondria. And after publication of the Nature paper, a very talented postdoctoral fellow who just joined my lab a few months before the beginning of the pandemic, Hongshu Xian, has worked out the entire pathway by which the oxidized mitochondrial DNA is chopped into little pieces. She identified the enzyme responsible for breaking down the DNA called FEN1. She has shown that FEN1 inhibitors can prevent activation of the inflammasome and inflammation. And then she has shown that the small fragments of oxidized mitochondrial DNA can leak outside of the mitochondria through different pores, one in the inner mitochondrial membrane, one in the outer mitochondrial membrane. They make it to the cytosol where they can bind NLRP3, and eventually they can leak out of the cell and find their way into the circulation. So having found that, we wanted to know what does circulating oxidized mitochondrial DNA does to the entire organism. So to study that, we carried out a very simple experiment. We injected mice with alum, which is used as an adjuvant for vaccines, and has been shown quite some time ago to be a very important activator of the NLRP3 inflammasome. In this case, we are not using alum as a vaccine adjuvant. We are using rather high amounts of alum, and we inject it into mice 3 or 4 times over the course of a month or 2. And then we found that the injection of alum resulted in not only the release of mitochondrial DNA, but also in formation of antibodies to double-stranded DNA that comes from the nucleus of the cells. And we have shown that the production of these antibodies was very much dependent on the release of DNA from mitochondria, because when we treated the mice with an enzyme called DNase1 that completely breaks down the DNA in the circulation, we inhibited the formation of these antibodies, which serve as a clinical sign for autoimmune diseases, especially lupus. So this is actually used in the clinic as a marker. And we have shown that the antibody production is related to formation of structures within the spleen called germinal centers, which are essentially little factories for the production of antibodies. And eventually, these antibodies, whose titer is quite high, get trapped in the kidney, and they clog up the renal tubules, and they lead to kidney inflammation or nephritis, which is part of very advanced lupus called lupus nephritis. And patients with this condition may end up needing kidney transplantation, because there's absolutely no way to reverse lupus nephritis, at least until very recently. So we asked, how does it work? And we have shown that, again, the enzyme OGG1, whose deficiency increases Alzheimer's disease risk, can block the formation of oxidized mitochondrial DNA, and blocks the formation of germinal centers, blocks the production of autoantibodies to nuclear DNA, and prevents the glomerulonephritis that is normally associated with lupus. To check what cells actually see the oxidized mitochondrial DNA, we use an analytical method called flow cytometry. We look at what cells are recruited to the site where we inject the alum, and we found that a cell type called plasmacytoid dendritic cells, or PDC, respond to the DNA and take up the DNA, and then that leads to NLRP3 inflammasome activation, caspase-1 activation within the plasmacytoid dendritic cells. If we treated the mice with antibodies that lead to depletion of plasmacytoid dendritic cells, we pretty much suppress the alum-induced autoimmune disease. And recently, antibodies that cause PDC, or plasmacytoid dendritic cell depletion, have been tested and have been approved for certain cases of lupus. So we think that all of this is relevant to the human disease. We have shown that once the plasmacytoid dendritic cells see the DNA, they are capable of programming another type of immune cell called T cells. These are CD4 positive T cells. We have CD4 positive and CD8 positive T cells. And what the PDC that have seen mitochondrial DNA can do is they convert what are naive CD4 T cells to a derivative of this cell type called follicular helper cells, or TFH. And TFH are very important for antibody production. And again, antibodies that reduce the number or deplete TFH have also shown promise against autoimmune diseases, including lupus. So we also use a genetic method to deplete follicular helper cells, knocking out a transcription factor called BCL6 that is very important for the generation and the maintenance of these cells. And again, that also protected the mice from alum-induced autoimmune disease. So to summarize what I've shown you so far, we find that in response to stress provided by the injection of alum or other substances, mitochondria undergo oxidative stress, their membrane is damaged, and they can then generate oxidized mitochondrial DNA that is broken down by the enzyme FEN1, can be released to the cytosol, and can also be released to the circulation until it meets plasmacytoid dendritic cells that are also present in the circulation. Once it's seen by the plasmacytoid dendritic cells, the oxidized mitochondrial DNA is taken up by the cells and again leads to activation of the NLRP3 inflammasome in the dendritic cells and induction of various cytokines in addition to IL-1 beta. And these cytokines are very important for the ability of the PDC, the activated PDC, to convert naive CD4 cells to follicular helper cells, which then act on B cells to stimulate the production of antibodies, including autoantibodies to nuclear DNA and other proteins that coat the nuclear DNA, which result in formation of nucleosomes. And all of this is very important for autoimmune disease. So now you will ask me, how is all of this related to Alzheimer's disease? Because what I showed you so far is very important for autoimmune diseases such as lupus. So the reason that we believe that this is important for Alzheimer's disease is that patients with Alzheimer's disease also show elevated amounts of oxidized mitochondrial DNA in the circulation. As I mentioned, loss of the enzyme OGG1 that is important for the repair of oxidized mitochondrial DNA greatly increase the risk of Alzheimer's disease. But we still have quite a lot of questions regarding the exact relationship between the NLRP3 inflammasome and Alzheimer's disease. So quite many papers have shown that somehow the NLRP3 inflammasome is related to Alzheimer's disease. Maybe it's activated by amyloid fibrils. and but exactly what activates it in the brain and what are the effectors that respond to activation of the NLRP3 inflammasome that are important for Alzheimer's disease pathogenesis is not known. And I want to add that the T cells, including follicular helper cells, which I mentioned earlier on, have been very recently implicated in Alzheimer's disease pathogenesis, but the mechanistic details are still nebulous. So to study exactly what's going on in Alzheimer's disease, we have used a mouse model of Alzheimer's disease called 5XFAD. These mice overproduce amyloid fibrils and also produce alpha-synuclein. These are two proteins that serve as markers of Alzheimer's disease. And also they are thought to be involved in Alzheimer's disease pathogenesis. Looking at production of oxidized mitochondrial DNA in microglia, which are cells that are very related to macrophages that we've been studying all along, but are present in the brain. We found that microglia from the Alzheimer's disease model mouse, not from normal mice, show interaction between oxidized mitochondrial DNA and NLRP3. And it's very clear that the NLRP3 inflammasome is being activated in microglia from these mice. And if we overexpress OGG1 in these Alzheimer's disease model mice, it completely prevents the generation of oxidized mitochondrial DNA and its binding to NLRP3. So if we had drugs that somehow can increase the production of OGG1, we can probably protect the mice and maybe even humans from Alzheimer's disease. But such drugs are very difficult to come up with, and probably we will never have those. So we and others have been focusing on identifying effective inhibitors for the NLRP3 inflammasome, for the enzyme FEN1 that is involved in the processing of oxidized mitochondrial DNA, and so on. But now I'd like to show you something which we think is very exciting and provides us with another avenue for either the prevention or the treatment of at least early Alzheimer's disease. So knowing that oxidized mitochondrial DNA can activate dendritic cells, and the dendritic cells can lead to conversion of naive CD4 cells into follicular helper cells. We have used flow cytometry to look at the presence of follicular helper cells in the brain and periphery and the spleen of the Alzheimer's disease models, the 5XFAD mouse. And you can see that in comparison to wild type, to healthy mice, the 5XFAD mice have more follicular helper cells in the brain. When you express OGG1, you completely prevent the accumulation of these cells in the brain. They also have more follicular helper cells in the periphery, which indicating that even though the 5XFAD mutation works to increase neurodegeneration, there are also peripheral effects due to the increase in circulating follicular helper cells, and some of these cells are present in the spleen. So once we found that, we were interested to know whether anybody else has seen the same thing and whether they found the same thing in human brains from Alzheimer disease patients, which are not so easy to come by. And that's why we conduct all the experiments with mice. And we also looked, before looking at the literature, at the nature of the follicular helper cells in the brain. And then we found that they produce a number of pro-inflammatory cytokines. They produce high amounts of tumor necrosis factor, TNF, a cytokine called IL-17, and also produce high amounts of interferon gamma. So we looked at the literature and we found a very exciting paper published about 4 years ago from a group at UC Irvine, with whom we are trying to collaborate now, that have detected the presence of follicular helper cells in Alzheimer's disease brains. And they have shown that the presence of these cells is linked to increased production of autoantibodies and increased production of inflammatory cytokines. And exactly whether it's the cytokines that are produced by the follicular helper cells or the production of autoantibodies that drives the pathogenesis of Alzheimer's disease remains to be determined. It's very difficult to do mechanistic experiments with human brain. So we think that our mouse system will provide many of these answers. And once we know what's going on, we can identify what is actually going on in the brain of Alzheimer's disease patients. So we postulate that the accumulation of follicular helper cells in the brain of patients with Alzheimer's disease is an important component of Alzheimer's disease pathogenesis. And in addition to establishing the role of these cells in the onset of Alzheimer's disease, we need to determine whether they act by secreting inflammatory cytokines or by stimulating the production of pathogenic antibodies. And this would depend also looking back at human specimens and see whether there are germinal center like structures that are responsible for the formation of autoantibodies in the brain or in the spleen of these patients, which nobody actually ever looks at. So there's still a lot of work to be done, but I think we are definitely making steady progress towards understanding of this disease. Before I end, I'd like to add that together with Pfizer, which is a very large pharmaceutical company, I was involved in the development of antibodies that can deplete both follicular helper cells and plasmacytoid dendritic cells. These antibodies are now in phase 2 clinical trial for autoimmune diseases, including lupus, and hopefully they make it through the next stage, the phase 3 clinical trial, and get approved if they are found effective for the treatment of autoimmune diseases. I would certainly recommend Pfizer to start looking whether the same antibodies can ameliorate Alzheimer's disease or even prevent the progression of the disease in early onset patients. Last but not least, I'd like to acknowledge the people who worked in my lab on this project. Much of the work that serves as the basis for the funding from the CART fund was done by Zhenyu Zhang, who's now an associate professor at UT Southwestern in Dallas. And he was assisted by Elsa Sanchez Lopez, who's now an assistant professor in orthopedic surgery at UCSD. And the most recent work in my own lab was done by Hongshu Xian, very talented postdoctoral fellow who just got a job at in Medical College of Wisconsin. So she's about to leave us. And she's been working with a Japanese scientist in my lab, Kosuke Watari. Again, thank you very much for your support, and I'll be happy to entertain questions.
Greg, go ahead.
All right. I was just, Dr. Karin, thank you so much for your time and for your presentation today. I know most of it goes over my head, but I was going to ask, back in 2017, you got $150,000 to study a particular theory from CART. How important has that been in your advancement of your understanding of all of this interconnectivity?
Okay, so the exact funds were to look at the involvement of CMPK2, cytosine monophosphate kinase 2, in activation of the NLRP3 inflammasome and eventually in Alzheimer's disease. So we have used the funds to generate CMPK2 deficient mice. We have shown that these mice are protected from activation of the NLRP3 inflammasome in response to different challenges. Elsa took the mice with her to her own lab because she was involved in their generation, and she is continuing to study the relationship between CMPK2 and inflamma aging. It seems that the mice don't age as rapidly as the mice that express CMPK2, but to study aging in mice, even though old mice are about 2 years old compared to 80 years old in humans, it still takes quite a lot of time and quite a lot of mouse breeding to generate the numbers of mice that we need for such studies. Thank you for your question.
Anyone else have questions while we have him today? Bill.
I just have a comment. As we go down the road to and giving out grants, it's amazing how simplistic the first couple of grants were from 1999 on, where we were focusing on big, broad spectrum events, thinking that a drug could come along and resolve it. And as we go on, looking at the grants that were given out this year, it is absolutely incredible how deep down we've had to go and how absolutely complex the issue is. And it continues to be complex and mystifying. I am absolutely amazed at how hard it is to try and figure out how to resolve Alzheimer's disease. And I'm also amazed at the spin-offs that have been associated with findings that we have funded that are leading to discoveries in other areas and successes in areas beyond Alzheimer's disease. But Dr. Khan, that just was amazing. And thank you for that. And thank you for recognizing your fellow researchers. You have an incredible lab, and hang in there. You're going to find a solution not only to Alzheimer's disease, but to lupus, to cancer, to all sorts of other stuff. So thank you from the bottom of our heart for what you're doing, and keep doing it. Thank you.
Yeah, actually, thank you so much for your support, but I like to make a comment. It's the nature of Alzheimer's disease because the disease happens in the brain, and the only brains that are available to us are from post-mortem brains. So we cannot go and do a brain biopsy, the same way that we can do cancer biopsy and get material even before the cancer advances and kill the patient. And then, with cancer, you also have surgery, so that gives you material and so on. And there's a lot of mouse models for studying cancer and so on. Autoimmune diseases are a bit more complicated because what people look at is the blood from the patients, and we are not looking at their spleen. Again, it's very difficult to go into somebody and take a piece of their spleen unless they're dead. But it seems that since the autoimmune diseases are due to overproduction of autoantibodies by B cells that are present in the circulation, it still provides us with ways to look at them. The problem with Alzheimer is we still don't know, after, I would say, 50 years of intensive research, whether amyloid beta, tau, alpha-synuclein, all these proteins that are overproduced and they leak out and present in Alzheimer's disease brains are actually a sign of the disease, or whether they drive the disease initiation or disease progression. We have seen now antibodies to A beta being approved as therapeutics, but it seems that they're not very effective. The approval process was largely politically motivated, and in Europe, the approval of these antibodies have been rescinded, so they are no longer approved for human treatment, and so on. And I think we really need to look at some new theories, at some new players, and hopefully looking at immune cells in the brain would lead to some progress, but I cannot promise that the progress will be quick.
Well, this is fascinating. Thank you so much. We are proud of our association with all of our researchers and very grateful that even after all these years, you are willing to spend some time with us and share your updated thoughts. It is always great for us to have that opportunity, and we do appreciate your time today.
Thank you so much. And I will also recommend some of the people who trained in my lab to consider applying for your support. So
Wonderful.
thank you again, and have a nice day, and the rest